Hey there, folks, and welcome to yet another COVID literature review. I am your host, Regretna, and today we're going to be looking at a really interesting paper that was just published in the Lancet a couple of days ago. Now, before we go too much further, as I'm always want to say, I'm not a medical professional. This isn't medical advice. I'm just a scientifically literate person with a camera and a microphone on the internet. So, take what I say with a grain of salt. But with that out of the way, let's hop into the paper. So this is the paper we're going to be looking at, Fast Evolution of SARS-CoV-2 BA.2.86 to JN.1 under heavy immune pressure. A couple of things in case you're like, what is going on? JN.1 was recently listed as a variant of interest by the WHO, so it's receiving a lot of attention right now. I will just mention that we have been paying attention to JN.1 for almost a month now. And I want to highlight this paper, which I'll post down in the description. This is a preprint, so it hasn't been peer-reviewed yet, but the G2P Japan research group is really good. So I'm sure that this paper is going to be published. This goes into a little bit more depth than the one that we're going to look at, which is just a letter. So it's a little bit shorter and conciser, but that makes it good for our purposes. So JN.1 is a variant that has descended from BA.2.86, and the introduction does a nice job of kind of laying out some of the natural history of what's going on here. So BA.2.86, when it emerged, garnered a lot of attention because people were worried that it was going to be very immune evasive, and it turned out not to. BA.2.86 did not show substantial humoral immune escape and growth advantage compared with current dominant variants, such as these two listed here. It showed remarkably high ACE2 binding affinity, and I really want you to remember this part because this is going to come back at the end of the paper. And we've talked about ACE2. In fact, if you watched the previous yet another COVID literature review, the ACE2 receptor, it plays a huge role in SARS-CoV-2 and COVID-19 and probably long COVID as well. So if it seems like I'm making a big deal about this, you'll understand why in a bit. Anyway, this increased binding affinity, coupled with its distinct antigenicity, could enable BA.2.86 to accumulate immune-evasive mutations during low-level population transmission, akin to the previous evolution of these variants listed here. So again, not to put too fine of a point on this, but the authors don't say this, but I will. This is one of the reasons why we really, really need to be intentional about decreasing the spread of SARS-CoV-2, because every time it gets inside of one of us and makes us sick, or doesn't make us sick, because a lot of cases are still asymptomatic, it has opportunities to replicate incorrectly, which is a thing that viruses do when they're making copies of themselves. They make mistakes. These mistakes can lead to mutations, some of which will increase fitness. And indeed, the theory that's kind of being put forth by the authors here is that because of the fact that BA.2.86 is really, really, really good at binding to ACE2, that means that it's going to get into our cells when we're infected with it, and it's going to have a lot of opportunities to stumble on a good mutation, and that might be what happened here. A thorough investigation into the immune evasion capabilities of JN.1, particularly given its few additional mutations, is imperative. And again, this is another thing that we'll come back to later in the paper. Now, just quickly to give you some very easy to process data to kind of increase your intuition about JN.1, you can see these beige, the beige columns are JN.1, and you can see that just going from October 23rd to 30th, and remember, this was like almost 2 months ago already, but it was in France. So, where you are in the world in terms of your closeness to France. Anyway, you get the idea. Big increase over the course of a week. This is almost like a tripling of the percent of SARS-CoV-2 infections being JN.1. So it's very fit. It is infecting a lot of people, and that's part of the reason why the World Health Organization has designated it a variant of interest. So you get the idea. All right, so we're going to scroll past the rest of these because they're a little bit too dense for our purposes here. So into the discussion. So we first study the humoral immune evasion of JN.1, easy for me to say, using pseudovirus. This is a term to art that you will see quite often if you're reading these kinds of papers. Basically, they take plasma from patients and then they expose the virus to it and see how much of the virus gets neutralized by the plasma, because our plasma has our immune cells and stuff in it. So this study included 2 cohorts. There was one with 27 participants who had a post-vaccination XBB breakthrough infection, and then another with patients that were reinfected with XBB after a BA.5 or BF.7 breakthrough infection. Now, I've re-recorded this video like 5 or 6 times, and this is the part where it always falls apart. So for the sake of simplicity, I'm just going to say that what this basically means, what these 2 arms give us, is different patients with different past histories with SARS-CoV-2. So the type of antibodies that they have and all this sort of stuff. This gives us kind of a nice broad sampling, even though it's only 2 groups. It'd be great if it was like 50 or something, but this gives us a good way of sort of comparing the behavior in each group and seeing similarities and differences. Anyway, the authors state JN.1 displayed significant enhanced immune escape compared to BA.2.86. Now, it is important to remember, because JN.1 is pretty serious, and I think this is something we should all be paying attention to, but I want to just reiterate like what they're saying here. In the introduction of the paper, they stated that BA.2.86 isn't super immune evasive. It's very good at binding to ACE2, and that's important, as I have made clear. But it's not like super evasive. And that sort of makes sense because BA.2.86 has been around for a while, and it is not the dominant variant. And I don't know if it ever actually was, to be honest. I don't know that off the top of my head. So saying that JN.1 is significantly more immune escapey than BA.2.86 does not mean that, in like some objective measure that JN.1 is like super immune evasive. However, it is important to acknowledge that this very simple mutation that JN.1 picked up made a variant, BA.2.86, which wasn't that immune evasive, very immune evasive. So again, we really need to decrease the number of opportunities that SARS-CoV-2 is having to figure out ways to be clever, because it's bad for us. Anyway, so the paper goes on to say that there was a 2.1-fold decrease in neutralization titers among individuals with the reinfected XBB post all these variants, and a 1.1-fold decrease. Basically, again, for the sake of simplicity, this is saying that the amount of evasion was different depending on what your past sort of history was with SARS-CoV-2, like the different variants you'd been exposed to and stuff like this. But the top line takeaway from this is that JN.1 is very sneaky. And on top of that, whether you've had 3 or 4 mRNA vaccines, or a mixture of mRNA and maybe Novavax, or you've only had the primary series, but you've gotten COVID a bunch of times, JN.1 is going to be able to get past your defenses and make you sick. That's really the takeaway from this. Now, I want to also mention, this is slightly out of scope for this paper, but I think it is worth sort of reiterating. The vaccines do not prevent you from getting SARS-CoV-2. They just don't. What they do is decrease the odds of you having a severe illness, of getting severe COVID-19, which is good. But what's better is to not get infected in the first place. And having these lower neutralization titers increases the odds of you getting any type of infection with SARS-CoV-2. And again, because of the fact that a lot of different cell types in our bodies have ACE2 receptors, being infected with SARS-CoV-2, it's not really that great for you. Anyway, going back to the paper, this is particularly interesting. JN.1's plasma evasion surpassed that of competitive variants HV.1 and JD.1.1. HV.1 and JD.1.1 also showed significantly lower plasma neutralization titers compared with their parenteral strains after acquiring this mutation, these two mutations respectively, explaining their growth advantages. So what this is basically saying is that these variants are all telling us the same thing. There's these handful of mutations that when they happen, they make SARS-CoV-2 better at evading our immune systems, our pre-built up protection against them. And this, by the way, is another one of the reasons why SARS-CoV-2 is not just the flu. It is changing very, very rapidly, and it's infecting a lot of people. And it also is not seasonal. It doesn't really go away. We've had, waves all throughout the summer. Anyway, going back to the paper, again, they do some more explanation about how essentially it seems like L455 is implicated and has something to do with the lowered ACE2 affinity. So actually, I just kind of spoiled the punchline a little bit. So by using surface plasmon resonance, we found a notable reduction in ACE2 binding affinity for JN.1 receptor binding domain, indicating that its enhanced immune escape capabilities come at the expense of reduced ACE2 binding. And then again, there's a fairly technical figure that shows you that. Again, not going to go into dissociation constants today, but interesting, right? So there's this kind of trade-off that's occurring between being really good at attaching to the spike protein and being able to attach to the ACE2 receptor. But what's interesting is this last sentence here. So the L452R mutation of HV.1 didn't affect binding affinity. So it's important to acknowledge, basically, to kind of take a step back for a second. So in the introduction, they talk about how that high binding affinity could potentially allow BA.2.86 to acquire some immune-evasive mutations. And they mention here that sort of Faustian dilemma of trading affinity for immune escape isn't necessarily true. And there's a couple of other things that they mention at the end of the paper that also kind of feed into this. But again, they're calling out here that it's possible, and HV.1 did this, that you can acquire some of these immune-evasive mutations that, again, essentially make our immune systems less familiar with SARS-CoV-2. And you can acquire that mutation and still bind to ACE2 very strongly. And you have to imagine that if we ever got a strain that was, basically as good as BA.2.86 at binding to ACE2, but was as immune evasive as JN.1, that would not be great for us because it would increase the number of opportunities for it to build ever better mutations and continue to be very infectious. Anyway, not trying to catastrophize here. So anyway, they do a little bit of a discussion about where in the receptor binding. Again, we're not going to go that deep. There's a little bit more discussion about some pseudovirus neutralization assay to figure out what types of antibodies JN.1 is good at evading. But here at the end, in terms of therapeutic antibodies, SA55 retained its neutralizing efficacy against all examined variants, including JN.1. So what this is saying is that this SA55 antibody does still work against JN.1, so it's not completely different, which is good. Together, these findings suggest that L455S greatly enhanced JN.1's resistance to humoral immunity through compensation of BA.2.86's weakness to class 1 antibodies. So what this is saying is that BA.2.86, part of the reason why we are able to fight it off relatively easily is because of its weakness to class 1 antibodies, but JN.1 acquired this mutation, and that has allowed it to be less susceptible to these antibodies. And so that's kind of what's going on here. So this is really I should have led with this. This is the most important part. In summary, JN.1, by inheriting BA.2.86 antigenetic diversity and acquisition of L455S, rapidly achieved extensive resistance across the receptor binding domain class 1, 2, and 3 antibodies, and showed higher immune evasion compared with BA.2.86 and other resistant strains like HV.1 and JD.1.1 at the expense of reduced human ACE2 binding. So JN.1 is very fit and is as sneaky as some of the other strains that we've run into that, again, are also resistant to some of these antibodies. And it has done this by trading off some of its ACE2 binding affinity. And this last sentence here is, again, the thing that I would like to leave you with. Such strains could survive and transmit at low levels since their antigenetic difference would allow them to target distinct populations compared with dominant strains and have the potential to quickly accumulate highly immune evasive mutations at the cost of human ACE2 binding capabilities. So this paper actually tells us more about our response to SARS-CoV-2 than it does the specific JN.1 variant. And what it is showing us is that the more we allow SARS-CoV-2 to infect us, the more likely it is that a variant with high binding affinity and basically the characteristics that make it good at finding good mutations, it increases the odds that those variants like BA.2.86 are going to find a population, a group of people that it's able to infect, not make super sick. And because people aren't taking SARS-CoV-2 seriously anyway, those people will go out and infect other people who are also susceptible. And while the virus is chilling in those people, it's going to replicate and replicate and replicate, and it's going to find ever cleverer ways of evading our immune systems. So, with all that said, please wear a mask, get the latest vaccines, and stay safe. SARS-CoV-2 is out there.